today's ESOF session titled Rethinking Vaccines. This session is organized by the European Research Council, an agency established by the European Commission to fund excellence in research. I am Giovanni Torrisi, and I work as Scientific Officer for the Science and Social Science and Humanities Unit at the European Research Council. With my colleague Clelia Salpea, who works at the Life Science Unit, we are the organizers of this ESOF section on rethinking vaccines. We'll have the pleasure to hear today 3 ERC grantees who are at the forefront of the research on vaccines from different disciplines. The first speaker is Jason Reffler. He's a professor of political science at the University of Exeter in the United Kingdom. He has an ERC grant titled The Bunker, the problem of European misperceptions in politics, health and science, causes, consequences, and the search for solutions. Jason will be followed by Professor Rino Rappuoli. He's a chief scientist and head external research and development at the GSK Vaccines, based in Siena, Italy, and researcher at the Toscana Life Science Foundations for his ERC grant. He is as well professor of vaccine research at the Imperial College in London. His ERC advanced grant is titled Vaccines as a Remedy for Antimicrobial Resistant and Bacterial Infections. Our third speaker is going to be Professor Kristin Stabel-Ben. She's professor in global health at the University of Southern Denmark, and she has a starting grant titled CHILIC, Child Health Interactions in Low-Income Countries. Other 2 persons will help us during our session on rethinking vaccines. Barbara Gallavotti is a scientific journalist who will moderate the second part of the session. And Serena Zacchigna. She's group leader at cardiovascular biology at the International Center for Genetic Engineering and Biotechnology in Trieste. She will be our host in Trieste in the site room. Now it is my pleasure to give the floor to my colleague Clelia Salpea for a scientific introduction of this session. Thank you.
Thank you, Giovanni. Our session's goal is to make you think vaccines in a different way. Vaccines are the most remarkable health intervention of the past 2 centuries and have led to the eradication of a number of severe infectious diseases, having drastically contributed to the increased life expectancy. Vaccines may also offer solutions to new major global health threats, such as antimicrobial resistance and the COVID-19 pandemic. I will take 1 minute more. However, the acceptance by society of vaccination as a preventive intervention is a sensitive issue, and sufficient immunization coverage remains challenging in many parts of the world. We have seen new outbreaks of diseases that were under control in the past in some countries because of vaccination hesitancy and mistrust in vaccines. Therefore, this session will address the expectation and needs of society from vaccines, as well as the hesitations and misperceptions of society about vaccines. We have today in our panel 3 ERC researchers who study vaccines and vaccination from different angles. We will start with Professor Reffler, who will explain why people are hesitant towards vaccination and how the relevant misperceptions are spread. Then we will continue with Professor Rappuoli, who will give us a flavor of his work at the front of research on vaccine development and how his pioneering method is used to develop modern vaccines. In the end, we will have Professor Ben, who studies vaccines from an epidemiological point of view, and she will explain to us the scientific data on the non-specific effects of vaccines. So let's start with our first speaker, Professor Reffler. The floor is yours.
Please, Jason, unmute yourself.
Hi, thank you very much. Sorry, I had to unmute myself. That was the little pause there. It's really a pleasure to be able to be here today and to talk about my work. And I want to talk about the reason public opinion about vaccines. And the public widely recognizes the value of vaccines. So over 3/4 of the public agrees with the statement that vaccinations are one of the most significant achievements in improving public health. So there's really widespread support for vaccines on this measure. At the same time, lots of members of the public have strong concerns about side effects that come from vaccines. So 41% of the public agrees with the statement, I'm concerned about serious side effects of vaccines. And so just under 1/3 of the public agrees to both of those statements, both recognizing that vaccines are an incredibly valuable aspect of public health, and at the same time being concerned about the side effects that might affect them or their families. And so it's this disconnect between belief about the value of vaccines and vaccinations and side effects that allow for misinformation and misperceptions to get into public debate, and that can have severe negative consequences for vaccination uptake. So here's a slide that shows the fraudulent paper, first authored by how I refer to him in presentations as he who must not be named, suggesting that there was a link between the MMR vaccine and autism. This is now one of the most researched areas in vaccine, and there is absolutely no link. Yet, following the publication of this article, this remains a persistent misperception in the public. Now, thankfully, it's gone down in recent years. And if you look at different age cohorts, the people who sort of came of age or entered child rearing age around the time that this was popular and before it had been fully debunked and retracted, they have higher levels of belief in this misperception than other people who are younger or older. But it's this concern about side effects that can have really strong negative impacts, and is one of the things that can sort of change people's attitudes about vaccines or their behavioral intent. There's some other factors related to vaccine hesitancy that would be much harder to change and that would be constant over time. So people who are broadly suspicious or distrusting of elites tend to also not support vaccination, but that's an attitude or a worldview that's probably much harder to change than discrete beliefs about vaccines. Now, that said, I'm now going to tell you about how it might be difficult to change discrete views about vaccines and the behaviors that you might care about, such as vaccination uptake. And I'm going to talk about 3 randomized controlled trials that we've done. The first was a study that we wanted to look at, is it possible to correct this underlying misperception that the MMR vaccine or vaccines in general cause autism? So I, along with some colleagues, designed an experiment in which we had a sample of parents, and we assigned a representative sample, and we assigned people to receive different information conditions. Some of our sample, we didn't give any information at all. We just asked them questions about vaccines and vaccination uptake. Other people, we gave different messages or different pieces of information to see how that would affect their views about vaccines. In one condition, we simply corrected this misperception. We gave people information that said, this link between MMR and autism is not true. It's fraudulent. This is one of the most studied areas in vaccine, and there's no evidence for this at all. We were doing this study with a physician at the University of Michigan, and he said, why don't we see if we can try and scare people into taking vaccines? So we also had 3 different versions of treatments that were designed to increase understanding of the danger of getting any of the 3 diseases of measles, mumps, or rubella. So we had one condition where we gave people information about the risks of the disease. We had another where there was a narrative of what somebody had experienced in terms of their child suffering from one of these diseases. And then we had another condition where we gave people images of what it looks like for children who have suffered from these diseases. And the good news here is that compared to the control, the people who got the corrective information improved in their belief accuracy about whether or not vaccines can cause autism. So giving people corrective information decreased the likelihood that they would agree that some vaccines cause autism. We can also see that the other 3 conditions, the risks, narrative, and images, those conditions that were designed to highlight concerns or the dangers of a disease, didn't decrease misperceptions. We wouldn't necessarily expect that. There's no information about the misperception there. Why would it do it? And this same, among people who were favorable towards vaccines, this corrective information also did not decrease people's willingness to give the MMR vaccine to a future child. But among the subset of the sample that was least favorable towards vaccines to begin with, giving people the corrective information decreased their reported intent to vaccinate a future child. And notice these disease risk conditions, the risks, the narrative, and the images, it has no effect. So even among the least favorable towards vaccines, trying to scare people into vaccine compliance doesn't have a noticeable positive effect. Now, we wanted to see if there was something interesting or unique about MMR, so we tried to replicate this study, and we looked at another common vaccine misperception, that the vaccine can give you the flu. And we found remarkably similar results, that giving people corrective information would correct the misperception, giving people information about the dangers of the disease or the flu did very little to change the misperception. And that, as you might expect, people who are more concerned about the side effects of vaccines on average are much more likely to believe that you can get the flu from the flu vaccine. But we find something similar, that among these high side effects concerned people, now this is a small segment of our overall sample, that here the correction again decreases future vaccination intent. There's no such negative effect for people who have low side effects concern. But again, we also don't see that the scaring people to compliance increases vaccine compliance. Now this raises questions about how do we think about having a vaccine for COVID-19? So while we're in the midst of a deadly global pandemic, and it seems like the only way out is through a vaccine. So we conducted an experiment, and I'm presenting one set of experiments. We've replicated this experiment in another country, and the results are almost identical. And so we asked respondents, as you may know, there's currently no vaccine for the coronavirus. If scientists are able to develop a new vaccine against the coronavirus, would you get the vaccine right away to protect yourself? Or would you wait to see if some people develop unexpected side effects? And what we see here is that same general pattern of there is a group of people that are very vaccine compliant. They say they will get the vaccine right away, about 33%. About half of the public, so they would wait to see if some people develop unexpected side effects. So again, we see this concern about side effects and concern about side effects leading to vaccine hesitancy and hesitation, but only very small portions of the population explicitly say they would not get the vaccine or that they don't know. So there really is very high levels of underlying vaccine support, even if it doesn't look like there's going to be a rush to take the COVID-19 vaccine right away. But I said this was an experiment. And so the thing that we looked at is one of the other things that we know has an effect on vaccine attitudes and vaccine compliance is attitudes towards pharmaceutical companies. People who have negative views of pharmaceutical companies also tend to have more negative views towards vaccines. So we wanted to see, does it matter who is seen as being responsible for creating a vaccine in terms of the public's willingness to take it up? So one half of our respondents got this version of the question, where we ask if scientists are able to develop a new vaccine. But then we asked a second version to the other half of the sample, in which we asked if pharmaceutical companies are able to develop a new vaccine against the coronavirus. Would you get the vaccine right away to protect yourself? And the distribution of responses is nearly identical. There's no significant difference between these two conditions. So I think that one of the positive things that comes out of this is that there is enough concern about COVID-19 for fairly obvious reasons that the public is not super concerned at the moment as who is developing vaccines. And so messaging isn't going to have to overly focus on who the creators of vaccines are, and that what people are really going to care about and what's going to determine sort of the long-term uptake of a COVID-19 vaccine is what its side effects profile and how this is communicated, and importantly, how public health messaging and key political figures are able to prevent bad actors to get into the public conversation, particularly with claims around exaggerated side effects, non-existent or side effects. So I think that there's a lot to be hopeful in terms of the public's broad willingness to take up a COVID-19 vaccine. And thanks very much for listening to what I have to say.
So thank you very much, Jason. Let's move to Professor Rapoli's speech.
Well, thank you. Thank you. It's a pleasure to be here today to talk to you about the project and about vaccines. And if I can have the first slide, I will start talking to you
Yes.
about vaccines. And
One second, please.
Yeah. While the slides come up, I will.
Yes, I've clicked, but I cannot do it.
Okay.
Yes.
Perfect. Thank you.
I'm sorry.
So if you can go to the next slide, this is a slide I like to use lately when I talk about vaccines. And the slide that per se has nothing to do with vaccines, it just tries to summarize in one slide the history of mankind, which starts a long time ago. Basically, 12 million years ago, our planet was populated by non-human primates. Then approximately 5 million years ago, the first individuals started to diverge and started the period of evolution of 5 million years that basically brought to Homo sapiens, which 100,000 years ago started to get out of Africa, met the Neanderthal, met the Denisovans, and eventually we came to our species. And in a period of really 5 million years. Now, the important part of this slide is this red line. The red line goes across the 5 million years of evolution, shows life expectancy. For 5 million years, our species had a life expectancy between 25 and 35 years. And then starting 1700, the life expectancy started to go up. You see at the end, the red line goes up, 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 up. And today, life expectancy in the Western world is 80 years or more. Bottom line, during the last 300 years, our species gained 55 years of life expectancy. And during the last century only, we gained 35 years of life expectancy. And obviously the question is, why in 5 million years, during so recently, all this change? Well, to address this question, I went back to some of the records in the United States, in this case, and asked the question, why were people dying in 1900, 1925, 1950? And in this slide, you see two colors, yellow and red. Well, yellow is mortality from infectious diseases. As you can see, in 1900, 55% of the people died from infectious diseases. And then the yellow decreased. Today, is very low, as you can see, but still very important, as we know today from the COVID situation. And most of the infectious diseases that used to kill people in 1900 are diseases for which that we don't remember anymore, like diphtheria. Diphtheria used to kill 40 million people, basically, and today, most of the people in this audience are pretty sure never heard of diphtheria, completely disappeared because of vaccination. And you can do measles, mumps, rubella, smallpox, all these kind of things. So clearly vaccines contributed to increase of life expectancy of our species. And if we live 80 years or longer today, is because we've been able to conquer infectious diseases. Today we live in the COVID situation. So a few days ago, we're talking to Luca Carra, and we said, well, I mean, what really, if we had a vaccine for COVID today, what would that mean? And we said, well, you know, that would mean liberty. That would mean that we will be free again. So the consequence of that is that basically infectious diseases do take away our freedom. And we saw that during the lockdown. We couldn't go out, we couldn't walk, we couldn't work, we couldn't travel, we couldn't meet friends, we couldn't visit relatives, grandparents could not see the grandchildren. And so if we had a vaccine for COVID today, or if we had a vaccine for COVID a few months ago, we would be more free. So I think vaccines are also able to give us freedom. So in the next slide, it will show the three things have been used to conquer infectious diseases: hygiene, vaccines, and antibiotics. Hygiene is extremely important. We saw this during the lockdown. We called the hygiene non-pharmacological interventions, because if you don't have vaccines, you don't have antibiotics, then you can only have quarantine, washing your hands, and social distancing. We have seen they're extremely effective, but I mean, the prices can be very high. The other thing is antibiotics for bacteria or drugs for viruses. And those are extremely important for bacteria. And we know when we are sick, we need antibiotics. The problem with them is that they generate resistance. So if you see at some point, the first antibiotic is used, and then you have resistance, and then you need another antibiotic. So from the first one, R1, you go to R2, another antibiotic. But then you get resistance, and then you need a third one, and then a fourth one. And then you may end up where we are today, that we don't have new antibiotics, and now we are in trouble because there is a lot of antibiotic problem resistance. On the other hand, with vaccines, when we have a vaccine, usually that takes care of the disease and may eradicate it, or may control it for a long time with no resistance. Indeed, in the next slide, you will see the history of all vaccines and all antibiotics and drugs. You see that for antibiotics, every few years you need a new one, a new one, a new one, a new one, because all the others, you get resistance that become obsolete. The same is true for drugs for anti-malaria. While for vaccines, when the vaccines conquer a disease, they conquer a disease forever. So that's why I like vaccines, because they free us from diseases, and they do it in a remarkable way. Now, one of the consequences I was saying before is that by using antibiotics, we have generated a lot of resistant bacteria. And this has become, I would say, probably the next big pandemic is going to be antimicrobial resistant bacteria. It's already coming, it's already present, but it's anticipated that in 10, 20 years will cause more death than cancer. And this is recognized by everybody, United Nations, the WHO, and you see in this slide, they recommend that we need to be careful about antimicrobial resistance. We need to act urgently. And obviously, for me, the way to address antimicrobial resistance is by vaccination. And the next slide shows that, fortunately, today we have a lot of tools to develop vaccines. We started like 20 years ago using the genomics to develop vaccines. Today we have the possibility to use human monoclonal antibodies from people who are convalescent from diseases and figure out which are the antigens that they are responsible for immunity, so you can develop good vaccines. And then on the right, you see you can use a lot of the new science, X-ray, ultraviolet microscopy, MMR, really to get information and design vaccines. So in order to make vaccines for antimicrobial resistant bacteria, I wrote my ERC grant, which you see in the next slide. Basically, and the ERC grant was basically to use the what I call reverse vaccinology 2.0, basically to start from monoclonal antibodies against antibiotic-resistant bacteria, by taking the blood from the people, isolating B cells, and discovering which are the antigens that are recognized by the monoclonal antibodies that protect from the disease. And that was the idea, to discover novel vaccines for antimicrobial resistant bacteria. And of course, we got this ERC grant. We started working early in 2019, and by early 2020, we started to get the first nice results about antimicrobial resistant, monoclonal antibodies against bacteria-resistant to antibiotics when COVID came. So basically, we switched immediately all our expertise from making human monoclonal antibodies for bacteria to COVID. And the next slide is a summary. The next one, please, is a summary of what we did in less than 3 months. Basically, we isolated, we took blood from 14 people. We isolated 4,200 memory B cells for specific for the spike protein of COVID. We found 453 monoclonals that were able to neutralize the virus. We were able to express 220 of them. And then we looked at their potency. And we found that many of them were potent, but not that potent. And so 65% of them neutralized the virus, half a microgram. But eventually, we found some very rare antibodies that neutralize the virus below 10 nanograms per ml, which means that they're extremely potent and they can be used for prevention or therapy of COVID-19 and also for better design of vaccines. So basically, we started using these monoclonal antibodies. We took the best ones, selected for development. We started industrial production of these antibodies, and we plan to be in clinical trials probably in 3-4 months. And the industrial development is ongoing right now. And here you see that we isolated many monoclonal antibodies able to neutralize the virus. And at the bottom, you see the extremely potent ones, the ones that neutralize the virus below 10 nanograms per ml, and they're extremely important. So right now we are in the position basically to develop, we are in the middle of industrial production of these antibodies, and we will be able to provide, hopefully, tools to prevent and cure this disease. And we believe that human monoclonal antibodies will be the first drug for COVID-19, because small molecules or the other drugs will take much longer. So we really hope this will be available, maybe in 6 months, these things will be available for people. That's what we've done. Since my job usually is to do vaccines, so 2 slides about vaccines for COVID-19. I think you have seen that today we have more than 300 vaccines in development. How is that possible? It's possible because today to make vaccines, you don't need a virus anymore. And this, all you need is the internet, is the sequence of the genome of the virus that can be downloaded from the internet. And then you can use that sequence basically to design a synthetic gene, and that synthetic gene will be used to make vaccines. And the type of vaccines which are made, you will see in the next slide, which are basically 3 types of vaccines. Once you have the synthetic gene, you can make a synthetic vaccine made by RNA. And you know there are a lot of RNA vaccines. One is by Moderna, one is by BioNTech, one by Imperial College, and many others. And that's the fastest one because it's a synthetic gene and uses a synthetic vaccine. Or you can, the second thing is basically you take the synthetic gene, you put into a viral vector, and you can get a viral vector that delivers your gene. And there are many vaccines which are based on viral vectors. The most famous one is the one of Oxford, but there are many vaccines in China, in Europe, one in Italy by Reitera, and yesterday we got a publication in the Lancet of the one from Russia, also based on viral vectors. And then there are, now for RNA and viral vectors, these are very fast vaccines. They can be developed rapidly, but today we don't have much experience with these things. So I think they're, from the phase 1 data that we got, they look like they're promising, but they've never been used in millions of people. So when we start a large-scale vaccination, we need to be very careful about watching the safety and effectiveness of these vaccines. The third type of vaccines is the one that takes longer, is basically when you make express the spike protein in mammalian cells or in yeast or in tobacco plants, and then you add adjuvants, so you make that as a vaccine. That vaccine takes a little bit longer to go into humans, but once you have basically that is fine. And for these vaccines, we have a lot of experience, and these vaccines will come 2 or 3 months later, but will be produced at large scale, and we have a lot of confidence about their safety and effectiveness. So bottom line, thanks to the new technologies, I think we're going to have the first results about whether these vaccines work in the next, maybe by starting October, November. And by the spring of next year, we'll start to have large-scale number of vaccines available for a lot of people. And hopefully these vaccines, together with monoclonal antibodies, will free us and will free the world from the COVID-19. And with that, I thank you for your attention.
And we can move now to our last speaker, Professor Ben.
Thank you very much for the invitation to be here and to talk with you and vaccine-interested people this Saturday morning about how vaccines train the immune system in ways no one expected. First, a disclaimer here. I'm a mother of 2 kids who both received all the vaccines in the Danish vaccination program. I've spent part of the last 27 years in Africa studying the overall health effects of vaccines, and I'm a co-developer of the concept that I'm talking about today, the non-specific effects of vaccines, and as such, I'm obviously biased. But apart from that, I haven't got any other conflicts of interest. My research that I'll be presenting today is partly funded by the ERC, but also by a lot of other founders and to whom I'm all immensely grateful. According to the current paradigm for vaccines, a vaccine is a biological preparation that improves immunity to a particular disease, and that understanding guides how we evaluate and monitor vaccines, because that's done in relation to the vaccine disease that they are targeting. Unknown to most people, none of the currently used vaccines in the vaccination programs were ever assessed for their effect on overall health before they were introduced. The effect on overall health was based on extrapolation. So for instance, if we have a measles vaccine and it's 90% efficacious and measles was 10% of all deaths, then it was assumed or estimated that introduction of a measles vaccine would reduce overall mortality by 9%. Our research has contradicted that understanding and that paradigm. We have a field station in Africa, in Guinea-Bissau in West Africa, where we follow a large number of inhabitants with regard to deliveries, and we follow the babies with their vaccinations, infections, hospitalizations, and their vital status. This allows us to do what nobody else did before us, namely to assess the effect of vaccinations on overall mortality and overall morbidity. And when we started doing that, something very surprising emerged, because we could not at all predict the effect on overall health based on what we knew about the vaccine effects against the target diseases. For instance, for measles vaccine, we saw that the introduction of measles vaccine, which was anticipated to reduce overall mortality by maybe 10%, was associated with a more than 50% reduction in all-cause mortality, so much more than could be explained based on the prevention of measles infection. But for other vaccines, we didn't see the same anticipated effect on overall mortality. Even though some vaccines protected against the vaccine disease, the vaccinated child actually died at a higher rate than unvaccinated children. This led to the realization that this equation here is too simplistic. Something was missing. And that is what we have called the non-specific effects of vaccines. Vaccines and their effects on overall health is a sum of their protective effect against the vaccine disease and also their non-specific effects against other diseases. Before I tell you what we found, I want to remind you that there are two major categories of vaccines. So we have the live vaccines. They replicate in the body. They affect all parts of the immune system very broadly and efficiently, and usually you only need one dose to get a full protection. When a child is born, it's a beginner when it comes to battling infections. And it's like somebody who enters the tennis court for the first time. And a real infection is a very serious opponent that can beat you out of the court. And I like to think about the live vaccines as a tennis coach who makes you run around the court and prepares you for the meeting with a real opponent. Non-live vaccines, in contrast, are not able to cause disease in the host, and therefore they're not perceived as a threat by the immune system. And they often need to be given with a helper substance, an adjuvant, and they need to be given in several doses to create protection. I think of the non-live vaccines as a ball machine, where the ball keeps landing at the same place at the same speed and teaches you one particular stroke to perfection. Both types of vaccines can create specific immunity. You need a little more doses of the non-live vaccines, but in the end, you will get the specific immunity against the vaccine disease. But it turns out that they have very different non-specific effects. Here's what we found out. We observed that all the live vaccines reduced overall mortality much more than anticipated. As if training with the tennis coach teaches you some tricks that prepares you very well for many different types of opponents. Non-live vaccines, in contrast, do the opposite. While you get protected against the vaccine disease, it seems as if training with the ball machine makes you worse off when you meet new and other opponents and the ball starts hitting somewhere else than what you trained for. And for reasons we do not yet understand, this is particularly seen in the girls. These findings were first done in Guinea-Bissau, but have now been confirmed in many other low-income settings in Africa and in Southeast Asia. The findings in relation to non-live vaccines are, of course, extremely concerning and uncomfortable. But fortunately, there are things to do about it. For instance, if girls receive live vaccines after non-live vaccines, they do not longer have that same increased mortality. And I'll be happy to elaborate on this if times allow in the discussion. For now, I'll just give you one example of our findings in relation to BCG vaccine, which is the vaccine against tuberculosis. It's no longer used in most high-income settings, but still recommended at birth in tuberculosis endemic areas. With funding from ERC, we conducted randomized trials of BCG at birth to children with low birth weight who would normally only get a delayed BCG vaccine. And we could show consistently in these randomized trials that children randomized to receive BCG at birth rather than the usual delayed BCG vaccine had lower mortality. In fact, it added up to an almost 40% reduction in all-cause neonatal mortality. Children don't die from tuberculosis in the first month of life, and we could show that the reduction in mortality was due to reductions in septicemia and respiratory infections. So showing beyond any reasonable doubt that the effect of BCG vaccine on neonatal mortality was truly non-specific. In 2016, WHO reviewed the evidence for non-specific effects of BCG vaccine and other vaccines and concluded with regard to BCG that receipt of BCG was associated with a reduction in overall mortality that was much more than expected through the prevention of tuberculosis. And in a joint analysis of the 14 studies included, BCG was indeed associated with almost a halving of all-cause mortality. It may seem really strange that a vaccine can affect how the body reacts to subsequent or unrelated pathogens, but we are getting much closer now at understanding what is actually taking place. And I want to show you one piece of evidence here from an experiment we did with Dutch collaborators. We took Dutch medical students and randomized them to BCG vaccine or no BCG vaccine, and 4 weeks later, we gave them a yellow fever vaccine as a challenge. And we could show that BCG vaccine induced epigenetic modifications in the innate immune cells, leading them to produce more IL-1 beta, among others, and also other cytokines, which again led to reduced yellow fever viremia. So this is a proof of principle, a really important one, documenting that in this case, a bacterial vaccine can affect how you respond to a completely unrelated infection subsequently. So to sum up what I've shown you here for BCG vaccine, we have epidemiological studies, many of them showing that BCG is associated with reduced neonatal, child, and even adult mortality. And this is not explained by prevention of tuberculosis. This is a non-specific effect of BCG. And we have immunological studies starting to tell us how this takes place through training of the innate immune system and its cells. And this, of course, has led to speculations whether BCG can be used against COVID-19. And as many of you know, or may know, it is also now being tested in numerous randomized trials all around the world. So to sum up and just show you this slide again, we have found consistently that vaccines have these non-specific effects. I showed you the results with regard to BCG vaccine, or at least some of them, but we've seen now for 4 live vaccines and 6 non-live vaccines, very consistent results. The findings obviously challenge the current paradigm for vaccines. There are so many studies now that show that this paradigm is no longer tenable. There are so many studies documenting beyond belief that vaccines do not only have specific effects. I and others have suggested that it is now time to change the paradigm for vaccines and start seeing them not only as vaccines against specific diseases, but as stimulants of our immune system. And we've elaborated on that in a recent review, and I'll guide you in that direction if you're interested in reading more about it. The findings and the new paradigm open for a new understanding of vaccine and a new use of vaccines. For children, I believe we can reduce overall mortality tremendously by taking into account not only the specific, but also the non-specific effects of vaccines. And with live vaccines early or live vaccine last policies, we could reduce overall morbidity and mortality significantly. In the elderly, I think that we could use the live vaccines to combat the immune system weakening that they experience. And we might also use vaccines for animals to reduce morbidity, reduce the use of antibiotics, and thereby antimicrobial resistance. And finally, as I hinted on, we might already have at hand live vaccines, which can be used as stopgap vaccines until specific vaccines are available in the current pandemic situation and also in future pandemics. So with that, I'd like to thank you for your attention.
So I would like to thank our three speakers for their wonderful talks and for sharing with us such exciting information on vaccines. And I would like now to give the floor to Barbara Galavotti, who will moderate an interactive round-table discussion. Barbara.
Thank you. Hello, everybody. It's really nice to be here. Thanks for inviting me. So, yes, we heard a lot of very interesting things. I look forward to make questions about that to our speakers. But first of all, much of what we heard has been obtained thanks to the funding that have been provided from ERC. I think ERC is the most powerful tool that has been developed by the European Union to support science and fundamental science in particular. So the first question is for Professor Torrisi. Can you explain us exactly what ERC is and how it works, how it is possible that it can support such an interesting development in science?
Yes, thanks a lot, Barbara, for this question. The European Research Council, it's a very peculiar funding agency. It was established more than 10 years ago by the European Union, and in particular funds frontier-driven research. There are some qualities of the ERC which are peculiar to it. The first one is that it's a fully bottom-up research agency. This means that there are no predefined calls or predefined teams or subjects or disciplines that are defined a priori but by the politicians or by the politics. In the ERC funds any kind of scientific knowledge with the only caveat that this knowledge, this research should be frontier, should be excellent and frontier research. This means that we trust a lot the community of the researchers, the research community, in order to do their research and do what they do best. This means bottom-up research. A second element peculiar to the ERC that we don't find necessarily fundamental research or applied research or basic research. We find specifically frontier of knowledge, frontier research. This research should go beyond the state of the art of the research. It should be excellent research, both in the proposal that is presented and in the excellent researchers that are evaluated by it. And this is what makes ERC a unique endeavor in the funding agency research world.
Mainly about vaccines and tools against epidemic disease. This is the most applied thing you can probably figure out to have in this moment. We owe to ERC also very fundamental research. You might remember the first ever image of a black hole that we could enjoy about 1 year ago, a little bit more, in April 2019. And this is another example of what can be obtained thanks to the funding of ERC. So it's very difficult to make a list or a ranking, but please, Dr. Salpea, can you provide us some more examples of what have been possible thanks to ERC?
Sure, Barbara. As you mentioned, this is the beauty of curiosity-driven research funded by the ERC. It spans from the image of a black hole to vaccines. I will pick 3 examples of ERC projects, with the first being the last year's Nobel Prize winner in medicine, Sir Peter Ratcliffe. Professor Ratcliffe, with his colleagues, shed light on how cells adapt to changes in oxygen levels, which is a fundamental research question, but is key to fighting a large number of diseases. Another striking example is that of the Mini Brains project, where Dr. Knogblich developed 3-dimensional organoid culture systems derived from stem cells, which were called cerebral organoids. These cerebral organoids can be derived from patients' blood or skin cells and can be used as a tool for the discovery and the development of therapies for neurodegenerative diseases and developmental diseases of the brain. They offer a better model compared to animals and therefore can replace animal experiments, reducing the costs for drug development, but also reducing the animal use. And I would certainly add a third one, the example of another ERC researcher, Professor Courtine. Professor Courtine and his team aimed at generating the functions of the spinal cord after injury. So they managed to develop the so-called personalized neuroprosthetics, which recently they showed that they can lead immobile rats and even paralyzed monkeys to overcome their paralysis. So this innovation of neuroprosthetics produced by this ERC project may in the near future make paralyzed patients walk again. So these are examples of ERC projects driven by scientific curiosity, which have now produced results that could lead to very beneficial applications for the society. And these examples are mainly from the biomedical field. So I would like also to ask my colleague Giovanni Torrisi to give us a nice example of such an ERC project from the social sciences field.
Thank you, Clelia. There are lots of different projects. Of course, we've seen Professor Reifler, who has made this wonderful example of misconception and misperception about vaccines. But there are a lot of other projects. For instance, one about the Denovian man, which was discovered in the region in Asia by an ERC grantees, which would permit us to understand how our DNA is shaped through the human evolution. There are many others, but I'll just stop here.
Thank you. So let's approach again the main topic of this session. To explain why fundamental science is so important, it has been done a nice example. It has been said that you can never obtain a light bulb by improving the candles. To obtain light bulb, you need really a new perspective, and this kind of perspective can be provided only by fundamental science. So I would like to ask to Professor Rappuoli, can you explain us? You have many light bulbs in your carnet, monoclonal antibodies, different new kinds of vaccines, and so on. What led you to these results?
Well, the fundamental thing is basically to have the technology to do things. I mean, we live in an extraordinary period because during the last 20, 40 years has been a kind of continuous technological revolution in all areas. And these technological revolutions, basically, they go from genomics, from human immunology, all this, allow us basically to conquer diseases that we couldn't even think about doing before. And so basically and doing this kind of job is fantastic because you do vaccines, you do human monoclonal antibodies, you do what we call fundamental research, you discover new secrets of nature. At the same time, if you're lucky, if you're good, your discoveries change the life of many people. And I don't think you can have a better job than this.
So let's go back to monoclonal antibodies. You said that we are probably going to have them before effective vaccines. They are very exciting. A lot of researchers all over the world are trying to develop effective monoclonal antibodies, but still there are some concerns. If they don't work properly, they can even make the infection worse through mechanisms like the antibodies the derived enhancement, or maybe you can have more side effects. Can you tell us which are your main concerns about the effectiveness of monoclonal antibodies? Do you fear something?
So thank you for the question. The question is very important because there is a fear both for vaccines and for monoclonals that immunity for coronavirus generating antibodies actually could make the disease worse, because not for COVID, but for other diseases like dengue or respiratory syncytial virus, has been shown in the past that some type of monoclonal antibodies can make the disease worse. Now, personally, I'm not worried about this for COVID, because I believe the science is different. We know that the antibodies generated by vaccines and our monoclonal antibodies are extremely good in neutralizing the virus. So if they neutralize the virus, the disease cannot get worse. On the other hand, this is a concern, and we need to address that scientifically and be careful while we develop both vaccines and monoclonal antibodies. And this is being addressed by all the people developing vaccines. And obviously, we need to keep in mind for monoclonal antibodies. Now, for monoclonal antibodies, fortunately, we have one trick that solves the problem. And the trick is basically to remove we can engineer the antibodies, basically to remove that part that theoretically could make the antibody worse. So that's what we have done. We have removed the what we call the portion that binds the receptor of the constant region of the antibodies that will theoretically could make it worse. So basically, I'm very confident that our antibodies will only do the good things because we remove the portion that will do the bad part, which I don't think is a problem, but since there are many questions, I don't want to run any risk. And there is no time to do a lot of experiments because this pandemic is moving very fast. I think the best way is, from my point of view, was to remove even a theoretical risk so that we can only stay with the good things.
Thank you very much. Yes, there is no time. This is really the point. We know that vaccine and drugs, vaccine even more than drugs, work because they are very safe. But if in a future vaccine or in a future drugs, unexpected side effect can come out, then could be we could have a big problem, because not only the life of some people can be affected, but also the trust of the general population against towards science and medicine can be affected for decades. On the other hand, we are hearing that in order to hurry up, in some cases, somebody is even considering to approve drugs or vaccines even before that all the trial part is concluded for the best and is succeed. So my question is to Professor Reifler. Do you think as a society we are prepared for a step that could be decided because of the hurry, but could not be the better one, what we are used at?
So my real strong advice to anybody who needs to hear it is to not take any shortcuts on guaranteeing safety and efficacy. while there is incredible immediate need for a vaccine, the long-term consequences of getting it wrong for COVID and for vaccines more generally are very severe. And this is absolutely not a place to take any shortcuts or unnecessarily hurry. I really can't express how grave I think the consequences would be of letting a untested or not fully vetted vaccine out. It might be okay, but if it's not okay, there'll be huge problems. But I also think that the not having it completely vetted opens up the opportunity for malicious bad actors with a anti-vaccine agenda to get into the space and to blame anything that comes up on vaccines, or to attack vaccine regulatory environment and approval environment. And yeah, so I think that as a society, given COVID-19, we're prepared to accept documented risks from well-tested vaccines if the risks and side effects are well communicated. I don't think that we're especially well equipped to deal with the long-term consequences of a rushed vaccine.
Thank you. Professor Ben, so we look forward for a vaccine directed against SARS-CoV-2, of course, a safe one, but in the meantime, we heard also from you that other vaccines might have an effect against it, existing vaccines. Then we heard that we might have some protection due to other kind of coronaviruses we met in the past in our life. But the mechanism why this past coronavirus might protect us is not very, very clear. It might it be something similar to the mechanism you are trying to figure out for the vaccines that have a broader protection than expected?
I think if we have this kind of cross protection that you're talking about from previous coronaviruses to the current one, it's presumably taking place through some kind of T-cell cross-reactivity where the two coronaviruses, or the previous and the new one, are so similar, so the T-cells simply think, well, I have seen you before, I know how to respond to you, and that's what seems protective. That kind of T-cell cross-reactivity may also be part of why we see these non-specific effects. I wouldn't exclude that. And for instance, it has just been suggested that BCG vaccine actually shares some homology with SARS coronavirus too. So it could be that part of the presumed protection of BCG against SARS coronavirus is the same kind of, hey, I know you, I've seen you before type of reaction. But generally speaking, I think we do believe, based on the current evidence, that the mechanisms that are most predominant in the non-specific effects of vaccines are related to this innate immune training. It's the vaccines that are modifying our first line of defense against pathogens, and that's really what makes the non-specific effects of vaccines so interesting in relation to the current pandemic and emerging pandemics, because it's simply a general strengthening of your first line of defense, which can be protective against a very broad range of infectious pathogens.
Thanks. So one of the effects of this pandemic, I would say one of the, maybe the only good side, is this, that it has pushed research ahead, especially in the field of vaccines. We had an unprecedented explosions of ideas and of also resource for experimentations. Still, it has been speculated that although the vaccines of new conceivment, like those based on RNA or DNA, are probably going to arrive first, and definitively in the future, they will probably help us to fight much better a broader range of diseases, still, probably the vaccine that will arrive later, because need a longer experimentation and a longer preparation, the more classical conceived vaccines will be more effective. This has been also, this point has been also raised by the New York Times recently. Professor Rapoli, what do you think?
Well, I believe this is true. I think what we know is that viral vectors and synthetic RNA vaccines are wonderful technologies, and they were at the forefront of research. But under normal circumstances, they were going to reach the market probably in 10 years from now, because you need the validation, testing in big, large trials. We don't have it in the industrial process. The vaccines need to be stored at -80, very inconvenient. So the technology is very primitive. It's been pushed to the limits by COVID, and we may have vaccines at the end of this year. And what we don't know is that we never tested these vaccines in millions of people. So we need to be careful, do pharmacovigilance, look at them carefully, and maybe everything is okay, but we need to be aware that we don't have experience about their safety and about their effectiveness. And I mean, frankly, I believe they will be okay. But, you know, until it's done, it's not done. So we need to watch carefully. And the world is aware about that. The other vaccines, the one based on adjuvants and protein, those, we have plenty of those vaccines. We have many of those. We know how they work. We know how to make them at the industrial level. We have plans to produce them. So all this industrialization, the possible side effects, all these things are well known. So we are very comfortable with those because we know they are safe and effective. So, and we know that they induce, in terms of antibodies, they seem to induce more higher titers of antibodies, which probably will provide longer duration of protection. So that's why we believe that the more classical vaccines will be kind of the, if you like, the second generation coming a few months later, but will be the ones we have confidence that will be working. In any case, for all these vaccines, we have seen extreme acceleration. You know, usually, I mean, to make a vaccine, I've done several, and usually it takes 15, 20 years from research to bring that to the market. And now we are doing in 1 year. So the acceleration is extreme. And I think in this acceleration, I think there are two things that you can put at risk that in theory. One is the financial investment, because you do the next investment before you know the result of the first one. And the other one is basically the safety. Now, in proceeding, I think there is an understanding of all the people who are developing vaccines and regulatory agencies, is that we should not take any risk in terms of safety and effectiveness, while we should take all the risks that we can about money. I mean, if we put money at risk, that's okay. I mean, this pandemic is costing us so much. So most of this acceleration is about taking a financial risk, but not taking a safety risk.
Thank you very much. Let's go back to our immune system. I still think it is probably the most complex thing ever developed by evolution. We don't really know in details how it works. The fact that vaccine might protect us from diseases different from those they were designed for is already surprising. Then I think you're also studying another very interesting aspect, which is the effect of vitamins. Can you explain us something about that? Dr. Ben, sorry.
Thank you very much for that question. And I very much agree with you. The immune system is wonderfully complex. We used to think it was more simple. It looked at a disease organism and responded to it and learned nothing but just about that specific disease organism. It didn't learn about other disease organisms in the process. But now it turns out, and perhaps it's not surprising from an evolutionary point of view that it is so, that in fact the immune system is much smarter. It's more like a brain. It learns from experience, and it can translate that experience into other unrelated situations. So it can really be learning and trained, and it can also sometimes be misdirected, just like the brain. And for that reason, all interventions that affect the immune system can interact with each other in surprising ways. And we found out that vitamin A supplementation given in high doses as recommended to children in Africa, that enhances the current state of the immune system. And that makes it very relevant what is going on in the immune system at the time when you give vitamin A. And it turned out that the effect of vitamin A differed very much depending on which vaccine you gave it with. So if you gave it with live vaccines, it was extremely beneficial, even in the absence of vitamin A deficiency, where it shouldn't have had an impact. But on the other hand, if you gave vitamin A with non-live vaccines, it was harmful even to children who were vitamin A deficient and would have benefited from the vitamin A. So the effect of any intervention needs to be seen in the context of the immune system, the complex immune system, and the effect of other interventions. And that's, of course, a terrible message because that's complicated. But if we ignore this complexity of the immune system and just think of interventions like vaccines and vitamins like isolated interventions, then I'm sure we will most certainly make some gross mistakes. But on the other hand, if we start understanding better the immune system and how we can turn it on and turn it off with our different interventions, then, as I also mentioned in my presentation, then there's a great potential for improving health globally.
Yeah, indeed, complexity is probably the key word for all our problems and all the problems that are provided by a modern and advanced society. But indeed, it's strange because we still have a lot of anti-science supporters. So science has been really successful in changing our life, our everyday life, but it seems it failed to change our mind. Other ideas, other philosophy can do it. Philosophy, for instance, religious preaching, even a political ideology for a while can change the human mind in a way that seems to be more effective than science. Science is perceived as cold very often, although it is probably one of the most romantic things that human beings can try, one of the most romantic adventures. So what is the problem? Professor Reifler, do you have an idea? Professor Reifler?
Yes, there are a number of factors as to why people have hesitation about scientific beliefs, and the exact rationale is going to change from which specific thing that we are looking at. So when we're talking about vaccines, the reluctance or vaccine hesitancy tends to be rooted in underlying causal factors like broad distrust of authority or elites, or in disgust sensitivity, so being particularly concerned about having something injected into your body. One of the things that's a positive about vaccine and vaccine misperceptions is that for the most part, there aren't strong political differences. That vaccination is not a very broadly politicized topic. It may be contentious in some quarters, but there aren't really strong partisan divisions, and there aren't really strong from mainstream parties really distinctive different messaging about vaccines. And I think that, which is different than, say, climate change, where particularly in some countries, the United States, Australia, United Kingdom, there's much stronger partisan and politicized messages about climate change. And so as somebody who's a political scientist, my great fear of what would make support for vaccination worse is if vaccines were to become highly politicized as a partisan issue, with one side being pro-vaccine and another side being anti-vaccine. But thankfully, we're not there yet. And hopefully we'll never will be.
Hopefully, yes, we hope so. So thank you very much. And now I would like to leave the floor to Professor Serena Zacchigna, who is in Trieste, and who probably already collect questions from the public, because I'm sure there are many questions to your topics. Thank you very much, Serena.
Everybody, welcome to Trieste, the current European capital of science, where the Euroscience Open Forum is ongoing. So we are all excited about these topics. Right, thank you. So we have a real public here, so anybody willing to ask questions, provide comments to our excellent speakers is invited to approach the microphone, and you can just say what you think of what you would like to know. In the meantime, I have already collected some questions from the online audience. So first questions, for instance, is for Professor Byrne, who show a higher mortality in females with non-live vaccines, which could sound counterintuitive. So can you elaborate a little bit more on that aspect? Could it be related with dimorphism in the immune response between males and females? Can you tell us a little bit more about?
Yes, I'm happy to elaborate. It is, of course, an uncomfortable finding, as I said, but we have seen it now consistently for 6 non-live vaccines that if we look at females who receive them, they have higher mortality overall than if they don't receive them. The findings are done in low-income countries, and they're done in the context where we do have, most of them are done in a context where we do have some herd immunity to the vaccine diseases, but nonetheless, none of the vaccines should have that danger signal of being associated with increased mortality. What further substantiates that this is a real effect is that it's not seen in boys. And if we compare vaccinated boys and girls with the non-live vaccines, meaning children whose parents all brought them for vaccination, then females have much higher mortality than males. And that is not seen for live vaccines. There it's actually the opposite, that females have lower mortality than males. So this is not just a question of sex preferential treatment. So the epidemiological data is quite clear, it's quite consistent, and it hasn't been contradicted. As I told you, none of the currently used vaccines were tested for their effect on overall mortality before they were being introduced. So we don't have any data to tell us that this cannot be true. There is actually some data to say the opposite. And furthermore, we have no known biases which could explain why it should be like this. Because for all we know, the biases in vaccine studies are the same all over the world. They favor the vaccine in such types of comparisons because parents are very reluctant to vaccinate their ill children, creating an inherent bias in favor of any vaccine in these comparisons. So I have to say the epidemiological data is quite clear on this. They are also quite clear on the sex difference. There we haven't nailed why it's there, but we are working on it. And I think there are more and more indications that we know that males and females, adults differ with regard to their immune system. We have now more and more indications that boys and girls also differ in terms of their immune systems. So there are various clues to what could be the underlying mechanism.
Great, thanks. And there is another related question. So why aren't we thinking at a possible live vaccines for COVID? I mean, has anybody think at inactivating the virus and use it as a vaccine?
I'm not, I think that Dr. Rapparoli maybe can answer that more efficiently than I can. I think that the recent reports about also long COVID and so on maybe make people extra hesitant to make anything with a live virus in it, any vaccine with a live attenuated virus. But personally, that had been my first choice if I was going to develop a vaccine against COVID. But again, I'm not a producer. This might be better replied by somebody else involved in the production of vaccines.
So let's redirect the question to Professor Rapuoli. Maybe also had another one for him, because one of the person listening to this session asked what are the main manufacturer challenges that antibody-based vaccines can entail. So maybe you can answer to both questions.
Yeah. So the first question about why we're not making live vaccines is indeed there are some people who are making live vaccines. As I told you, there are approximately 300 vaccines being developed. The reason why they're not in the media, they're not front runners, is because to attenuate a virus takes time. And once you have attenuated, you need to be absolutely sure that this attenuated is not going to revert to be a pathogen and is going to be absolutely safe. Doing all these things takes years. So live attenuated vaccines could become available, and some of them are in development, but there will be, we'll have confidence in them only a few years from now. We don't have mechanisms to be fast like we are doing with non-living vaccines. So that's why we have RNA vaccines, that's why we have vector-based vaccines, which are the ones that mimic more closely the live attenuated. So that's the reason why we don't have live attenuated vaccines. About challenges of manufacturing. So when we talk about vaccines today, basically we talk about people say, well, we should vaccinate 8 billion people, which are the population of our planet. And most of the vaccines require at least 2 doses, so we'll need 16 billion doses of vaccines. Now, the vaccine industry globally produces probably 1 billion doses of vaccines per year. So going to 16 billion of only one vaccine is going to be a huge challenge. Some of the new technologies like DNA and viral vectors may allow for production of hundreds of millions, maybe 1 billion doses of vaccines, probably in 2021. But they've never been industrialized. So huge money is being invested in industrializing them. Probably will happen, but we will never know until we'll see whether we will be able to do that in such a large scale. For the protein-based plus adjuvants, I think those are vaccines for which capacity is already there in the world. So I think we'll be able to get probably 1 billion doses of vaccines by the end of 2021, and then 1 billion or 1.5 billion per year after that. Because the industry is basically ready or has already planned to produce those type of vaccines. So I think that is going to happen.
Thank you very much. Sorry.
Yeah, about antibodies, there is there is plenty of industrial capacity to produce antibodies because there are more than 100 human antibodies licensed for therapies. Most of the therapies are against cancer, against autoimmunity, against inflammation. So if needed, I think antibodies can be scaled up substantially. Obviously, antibodies, you never get the same amount of doses as you get for vaccines. So we'll never have billions of doses of antibodies, but you don't need billions of doses for therapy. So I think if needed, will be capacity to make antibodies.
Clear. Thank you very much. So one question also for Professor Reifler, asking him to be synthetic because we don't have a lot of time yet. And the question is, from your perspective, what could be the main political or policy-related issue that may even hamper or impulse the development of COVID-19 vaccines?
That's a great question, and asking an academic to be brief that's a big challenge. I think that what concerns me the most is the extent to which leaders engage in overhyping vaccines too early in their life cycle. Or before we have full proof of their safety and efficacy and longer term consequences. And I'm a little bit worried about a sort of international vaccine race where every country is trying to get their vaccine out there first, and is that the normal sort of cognitive biases that come once you feel that you are rooting for a particular side come into play, that people might be blinded to the strengths of vaccines from other countries or the weaknesses of a vaccine developed in their own country. And that's about as brief as I can be.
Thanks. I think that was perfect. And I would give the word back to Clelia, who would like to close the session.
Oh, I'm sorry, I was muted. Thank you very much, Serena. I think we ought to give the floor back to the three speakers to give us their take-home message before we close on vaccines. So let's start with Professor Reifler.
So my simple take-home message is that it's very hard to scare people into taking vaccines. And that a unfortunate positive of COVID-19 is that the crisis is sufficiently real and visible to people that I don't expect dramatically large amounts of vaccine hesitancy. The other thing that I want to point out, which is an important, not a takeaway from my talk, is that while I talked about some elements, how that there can be a backfire in specific populations, I wouldn't want anybody to take away the message that therefore it is pointless or a bad idea to try and counter misperceptions about vaccines, or to counter misperceptions more broadly. To the extent that the overall mix of information that people receive affects their beliefs, that having a good and constant source of correct information and corrections is important in the long term. So please don't take away the because there can be a backfire, therefore we should just give up and not try and correct misperceptions.
Thank you very much. And Professor Rapoli, what is your take-home message?
Well, kind of a couple of messages. One is basically the vaccines are one of the most important conquests of civilization, I believe, because thanks to vaccines, we conquer a lot of infectious diseases and we live longer. What we learned during the COVID is that not only vaccines can make us healthy and live longer, but they can also give us freedom. So vaccination gives us the freedom that we like to have. Freedom to walk, to not be locked down, to all these things. And the last message is, fortunately, thanks to the revolution in technology that we have, today we can do vaccines at unprecedented speed. And we can also make human monoclonal antibodies for therapy. And these two tools will become available shortly. And people should be confident that basically the scientific community and the international community will not take risks of safety and efficacy, and should be confident that we will basically get rid of this COVID thanks to vaccines and human monoclonals.
Thank you, an optimistic message. And Professor Ben, what is your take-home message?
I have my home message here on a slide I put up, and I hope you can see it, because I would be very glad if at least some in this audience would take this message that every time you hear or talk about a vaccine, try to ask, what is the overall health effect of this vaccine? And usually the reply will be a statement about its protective effects against the vaccine disease. But I hope I've convinced at least some of you that this may not be the same. So this is my key message.
Thank you everyone today for this wonderful session.